right? Let's move on to the second learning objective, which is to describe the physical and structural supports of the central nervous system. So what are the physical, tangible structures that are protecting the brain and spinal cord? First off, we have the bone, which is the skull. This is having the cranium and the vertebral column. The cranium being the skull portion, protecting the brain, and vertebral column being the spine portion, protecting the spinal cord. Within the bone, we also have these dural layers, or these meningeal layers, rather. So there's the dura mater, which is the thick, tough outer protective layer, the arachnoid mater, which is a thinner, more spidery sort of meshwork, almost like a fishnet, if you will. That's kind of how the arachnoid mater appears. That's why it's called the spider layer or the arachnoid mater. This is going to be having the cerebrospinal fluid within it. The cell's going to have arteries and blood vessels that are supplying the central nervous system. And then pia mater is going to be the innermost layer. This is the tender mother, very thin layer that is intimately connected with the brain and spinal cord, and it is essentially indistinguishable. It's one with the outer layers of the brain and spinal cord. So in between the pia mater and the arachnoid mater, this is called the subarachnoid space. And in between this space is where we have cerebrospinal fluid around the brain and the spinal cord. So here's the skull, right? The bone that is housing these delicate structures. And if you have the opportunity, I think you still, if the lab sequence is still the way that I remember, you may have already had the brain. Hopefully you've had, but you can lift the brain up. It's usually in that bucket of fluid. And when you hold it, you can notice how delicate that tissue is. Very sloshy, very sort of jelly-like. Usually when we fix it with formaldehyde, it will usually harden to some degree, but really understand how delicate it is when it's inside the body, inside the skull. So the need for the skull is to help protect that delicate tissue from any damage from moving around when you move your head around. And this is also the role of CSF. CSF helps us float it into that fluid, so it's suspended and it doesn't actually start smashing around against the harder skull, which forms that cranial cavity. Deep to the skull, we have the dura. Deep to that, we have the arachnoid mater, and then deep to that, we have the pia mater. Again, pia mater is essentially one with the brain and spinal cord. And then in between these two layers, between the purple layer and the yellow here, the pia, is where we have that CSF circulating all around the skull, the brain, and all around the spinal cord. We also want to identify the dural sinuses. So the dural sinuses are these collections where venous blood is in the around the skull and meninges. It is going to be communicated with the arachnoid villi. So these projections from the arachnoid mater. So here's the arachnoid mater, here's the pia mater. Now these villi actually project up into the dural sinuses, and this is how CSF can be reabsorbed back from the subarachnoid space into these dural sinuses. And so CSF pretty much circulates around the brain and spinal cord, gets reabsorbed by way of these arachnoid villi, and then is going to go back into the venous system and then go back into pretty much the veins and then back to the heart. So that is how CSF circulates. It's made by the ependymal cells lining the ventricle. It circulates into the subarachnoid space, and then it gets reabsorbed into the venous system by way of these sinuses. And then that process can happen over and over again so that it can be made, circulated, reabsorbed continually. Looking at a cross section here around the spine, we can see a similar makeup of these layers. So we have the dura mater, the blue layer, the arachnoid mater, and then the pia mater intimately covering the spinal cord. So if we think about CSF now, looking more closely at that, it's a watery, clear fluid that's bathing the central nervous system. We talked about its main role being to cushion the brain so that it's not moving against the hard bone and the bony surfaces. It's suspended in this interstitial fluid. But also it helps to make sure that the CNS is in its own interstitial fluid environment, right? It helps to create this unique fluid environment that is bathing the central nervous system, that has its own unique makeup of electrolytes, its own unique makeup of cells, of fluid, that can help to provide the unique needs of the central nervous system that differ from the interstitial fluid anywhere else in the body. Again, it's made by ependymal cells that form these structures called the choroid plexus. So these regions along the ventricle formed by these ependymal cells are called the choroid plexus. And then it's going to be reabsorbed back into the venous system by the arachnoid villi. Now, if you look at the composition of plasma and compare that to CSF, so here's plasma having lots of glucose, lots of proteins, sodium at 145 of sodium, millimeters of mercury of sodium, potassium, chloride, and then calcium. So for the most part, the electrolytes are pretty similar in the plasma as they are in the CSF, right? There's slight variation here, except for chloride, which is really high in CSF. However, glucose and proteins drastically differ, right? Drastically lower in CSF. So there's much fewer proteins in CSF and much fewer glucose in the CSF as well. And we'll talk about how this comes to be. This is really because of the blood-brain barrier. The blood-brain barrier creates these tight junctions where large, bulky molecules like proteins cannot readily cross that barrier and go into the CSF. This is just a look at the ventricular system. Again, I think you'll speak more about this in your lab, but just to review the structures that are forming these cavities where CSF is made before it's actually circulated to the subarachnoid space. So it's made in the ependymal cells. If you look really closely here along the lateral ventricles, you can see these pockets of cells that are lining this ventricular cavity. So this is forming the choroid plexus, which is made by the ependymal cells, one of our glial cells. So CSF is made right here, circulates into the lateral ventricles, which are these large chambers. There's one on the left, one on the right. Then it goes through this tiny opening and enters the third ventricle, which is right in the middle of the thalamus. The thalamus is going to be on either side of this third ventricle. And then it's going to go through another tiny opening called the cerebral aqueduct and make its way to the fourth ventricle. From there, it's going to go through the central canal and then eventually through other foramina that lead out into the subarachnoid space. And then it's going to ultimately circulate in the subarachnoid space and then be reabsorbed by the arachnoid villi. Okay, so that is how CSF is made, circulates through the ventricles, then into the subarachnoid space, and then eventually be absorbed continually. Okay, let's shift to our third lecture objective to talk about the nature and significance of the blood-brain barrier, the cells that form it, the function of that barrier, as well as some of the features that are formed at these junctions. Now, the blood supply to the central nervous system is very high, right? It's about 15% of the total blood supply in terms of our total cardiac output going to the CNS. So although the central nervous system applies for 2% in terms of total body weight, it receives a lot more, a much higher proportion of our total cardiac output. It also has a very high metabolic weight. So because our brain uses about 20% of oxygen at rest and about 50% of glucose at rest, it has a high metabolic demand, right? It needs a lot of glucose, needs a lot of oxygen, even while we are resting, okay? And so that's going to be determined by a steady flow. So the brain needs a steady supply of blood in terms of blood vessels coming to the brain. It's very highly vascularized. There are actually two blood vessel networks that supply the brain, forming what's called a circle of Willis, right? If you've ever heard of the circle of Willis, this is a network of blood vessels that supplies the brain from the anterior perspective and from the posterior perspective, just in case there's any blockage on either side, which tells us how reliant the brain is on its steady blood supply. Now, thinking about blood supply to the nervous system, neurons depend on aerobic glycolysis mainly. So they need oxygen in order to supply ATP for the central nervous, for the neurons. So this needs a steady supply of glucose and oxygen. Now, there's a small amount of glycogen that can be made. We talked about this coming from the astrocytes, but for the most part, there are not a lot of glycogen stores in the brain, right, in the CNS. So while these glial cells can make a small amount, the brain is, for the most part, heavily reliant on a new supply of glucose coming in to give it energy. Fatty acids can also be, fatty acids can, to a small extent, this has not been used for energy, but please make a note here, to a small extent, fatty acids can be used by way of making ketones. So we can break those fatty acids down, convert them into ketones, and that can be used under extreme conditions, okay? So this is where we have ketosis, for example, and we'll talk about this in the endocrine system. So if you have limited supply of glucose, your body can shift into using fatty acids to make ketones, which can be used in a similar way to glucose, less so in the central nervous system, but it can be a backup supply for extreme circumstances. Now, when we have occlusion of any of the blood vessels in the brain, this is going to result in a stroke. And there are two different ways that we can see a stroke. Either if a blood vessel is blocked, let's say you have a clot that has formed and it blocks the flow in one of the arteries in the brain, that's an occlusive stroke, versus a hemorrhagic stroke, which is usually seen in the setting of chronic hypertension or chronic high blood pressure, where there's weakening of the walls of the blood vessels in the brain, and that leads to rupture or bursting of those vessels, which leads to a hemorrhagic stroke. So we can have an occlusive stroke, which is formed by clots, or a hemorrhagic stroke, which is formed by a bleed, right, a blood vessel rupturing, and both of these lead to stroke. What are some stroke-like symptoms that we would expect to see? Because this is the brain and spinal cord, we would expect to see weakness, right? Weakness usually on one side of the body, depending on where that bleed or that clot is. We can also see a speech impediment, inability to speak, inability to see, inability to receive sensory stimulus, so numbness to certain parts of the body, cognitive decline, all types of varying symptoms can be seen depending on the severity, the length of time that the brain has gone without blood supply, as well as the type of symptoms, right? The part of the brain that is being blocked or there's a bleed. So all of those can determine exactly what type of symptoms we see, and there can be very small minor symptoms, right? Someone can have a stroke and just have maybe a small speech impediment to much more severe consequences, which can even lead to death. Looking at the blood-brain barrier more closely, we have capillaries that are forming this barrier between the interstitial fluid around the brain and the plasma that's actually inside the capillaries. So the capillaries is the site of exchange between these two mediums. And so there's a thin layer of endothelial cells that are tightly adhered to each other by these tight junctions. So there can be some diffusion for really small lipophilic substances can really diffuse right across. But for larger polar substances, those tight junctions block any type of paracellular movement or any type of larger molecules moving across. Now, the need for the blood-brain barrier is to limit the exchange between those two areas, between the interstitial fluid of the brain and the blood that is in the vessel, to limit the exchange between those two areas. And again, it's going to be formed in part by the foot processes of the astrocytes. So here is a blood vessel in any other part of the brain. Here's a typical capillary. And then here's a blood vessel in the central nervous system, right? So a brain capillary. Notice that we have one, the astrocytes. So the astrocytes, their foot processes are going to be along these junctions. So the junctions here are going to be guarded by the foot processes of these astrocytes, just ensuring that there is no transport between the blood and the interstitial fluid of the brain. Another feature that helps with the blood-brain barrier is the tight junctions between these endothelial cells. So anywhere else in the body, there are small pores between these between these endothelial cells, but in the brain, these are tightly adhered, right? Nothing can get through there. Tight junctions are going to very tightly adhere the opening. So here we can see a little gap, and here there's absolutely no gap, as well as the foot process, just adding another layer of surveillance to these capillaries. So notice that things like proteins, as well as hydrophilic solids can go right through this membrane. In the brain, hydrophobic solids can get through, right? Things that can get through the cell membrane can diffuse, but nothing can get right through. There are no open pores for that type of transport. Also, things like proteins obviously are large and bulky and cannot get through. Lectroductic 4. So we're going to talk about myelin, how myelin is made, and the two different cell types that make myelin in the central nervous system and the peripheral nervous system, respectively. So what is myelin? Myelin is essentially a sheath that is wrapped around the axons of neurons that helps to insulate these axons, that helps to speed up the action potential, right? Speed up the impulse conduction. Now, myelin is essentially fat, right? It's a fatty layer, if you really look at it closely. And so when we see it in the central nervous system, it's going to appear as white, right? This is why we refer to as, this is what we refer to as white matter. White matter is the appearance of that fatty layer of myelin covering over the axons of these neurons versus gray matter. Gray matter is the cell body. So this portion of the neuron is going to be forming gray matter, whereas the myelinated portions of the neuron are going to be forming white matter. All right. Now the configuration of these differ. In the central nerve, in the brain, gray matter is over top the white matter. Okay, we can kind of see that here. Gray matter, which are the cell bodies, is on top, forming kind of this blanket over top the white matter, which is the actual myelinated axons. Now, in the spinal cord, the orientation is reversed. In the spinal cord, the gray matter is inside in the middle, and the white matter is going to be on the outer aspects. All right. Also, we want to think about these fibers. So these fibers that are forming these tracks that run through the spinal cord, these are sending communications from different parts of the CNS, as well as from the PNS to the CNS. And so these tracks are also going to be the axons that are sort of bundled together, but also myelinated, that are running throughout the brain and spinal cord. Now, again, as we mentioned, oligodendrocytes myelinate in the central nervous system, and Schwann cells, moving on to look at the peripheral nervous system here, Schwann cells are going to be the cells that form myelin in peripheral nerves, right? Structures that are outside of the central nervous system. And the way that this is done is that there's a sheath that is formed called neurolemma. So here is a Schwann cell. Here's the nucleus of the Schwann cell. And then here are these sort of arm-like projections that are formed by the Schwann cell called the neurolemma. So the neurolemma wraps around the neuron. Here's the axon of the neuron in blue. And as it wraps around that axon, then it's going to start to create this layer in between the Schwann cell itself and the axon. And this is what actually lays down the myelin. So really important to distinguish the neurolemma from the myelin. The neurolemma is not the myelin. The Schwann cell actually forms a layer called the neurolemma. So this entire yellow region that projects around the axon is called the neurolemma. And then in between the axon and the Schwann cell, the myelin is produced that actually begins to insulate the axon. All right. So the neurolemma is actually the structure that helps lay down the myelin from the Schwann cell. We've also talked about the way that gaps are formed in this myelin sheath. So this is not a continuous sheath from soma to axon terminal. It actually has these gaps in between called nodes of Rodbere where myelin is absent. And this is where we have that concentration of voltage-gated sodium channels that help to propagate the action potential and help to insulate or speed up that impulse from the soma or the axon hillock, more specifically to the axon terminal. So here is what the myelin looks like in the peripheral nervous system. Here is an axon. Here's a myelinated axon. Here's the Schwann cell, right, which is kind of wrapped around the axon. And then here's an unmyelinated axon. So here is that the nucleus of that cell, the axon that is, and then here is the Schwann cell that is still tightly wrapped around the axon. So even in unmyelinated axons, we still have Schwann cells sort of engulfing them, and we still have neurolemma. So again, the neurolemma is not the myelin, because even in unmyelinated axons, we still have neurolemma, but the wrapping that is created, which we can see here, is absent in the unmyelinated